Hello and welcome to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really happy you're here today. So this is a first time for us. We're doing this out of a hotel room in Washington, D.C. And we will be talking about multifunctional ADCs with Jagus Junutula today. Welcome to the show, Jagus. We're glad to have you.
Thank you so much for Drug Hunter giving this opportunity to join this session. I'm looking forward to it.
And now, without further ado, I'd like to introduce our guest, Jagus. So Jagus Junutula, he's the co-founder, president, and CEO of Arvic Therapeutics, and has more than 20 years of oncology drug discovery experience and over 30 years of life science research experience. Jagus obtained his PhD from the Indian Institute of Science in Bangalore and conducted postdoctoral research at Stanford. Jagus' career at Antler A Therapeutics, MopMap Therapeutics, Cerelan Therapeutics, and Genentech spent over 20 years, and throughout this time, he was responsible and contributed to several INDs and IND-ready programs. His work focuses on the discovery and development of biologics, including the discovery and validation of ADCs, engineering of next-generation ADCs, and development of bispecific T-cell engaging antibodies. Jagus contributed to the discovery and development of two approved ADCs, Kadcycla and Polivy, and he developed a novel thiomap technology platform generating site-specific ADCs to improve the therapeutic utilities of antibodies. Jagus was also a featured inventor in the greatest hits in 20 years of Nature Biotechnology and biomedical research in 2016, and he has authored more than 60 peer-reviewed publications and is listed as inventor on more than 300 filed or issued patents. He received the long-standing contributions to the ADC Therapeutic Field Recognition Award in 2017 at the ADC World Summit in San Diego. Jagus also serves as the editorial board member of leading journals, for example, the ACS Medicinal Chemistry Letters, and he was an Alexander von Humboldt Fellow, and he's also the co-founder and board chairman at the Science Gurus, a nonprofit organization that supports the development of next-generation scientists. So welcome. We're so honored to have you today, Jagus. Really a pleasure to have you here.
Thank you, Dennis, for those kind words. I really appreciate and looking forward to talk to the forum.
Awesome. Yeah, and in the good old Drug Hunter fashion, we typically start with a few questions to get to know the speaker a little bit better. So Jagus, can you maybe talk a little bit to us about what inspired you to start a career in drug discovery?
Actually, to start with, I'm an academic scientist. So my industry transition happened accidentally. I joined postdoc at Stanford, and then my postdoc mentor, Richard Scheller, and decided to move to Genentech. And then he gave an opportunity for all the postdocs to join that year to come and join Genentech. And we just took that opportunity and moved on. And ever since I joined Genentech in 2001, I never looked back. So it was a very interesting journey, even though I thought I would become an academic professor. But once I joined Genentech, completely opened a new opportunity, a field like drug discovery. And ever since then, I never looked back. And I think I always look into this kind of thing. You don't know when something knocks on your door. You probably have to keep your eyes open and take on those opportunities. And they can only happen once in a lifetime. So I think I probably made one of the best decisions at the time. So.
Awesome. We talked at the pre-meeting just how many things we have in common, and that's one of the things we do have in common as well, Jagus. Yeah, so second question was, what is your favorite molecule and why?
My favorite molecule, always I'm biased, I would say Kadcyla, because that's what introduced me to the drug discovery. And thanks to, greatest mentors I had at Genentech who introduced me to the ADC platform, and Kadcyla was the first one I happened to touch it in multiple different angles. But believe me, I still work on it. So in different way. So how do we make better drug? Always HER2 or Herceptin, always my favorite. Any HER2 franchise I would call my favorite because I learned a lot. Entire drug discovery, and every day we use Herceptin. So I think I would call Herceptin, Kadcyla, Enhertu, Pertuzumab, Perjeta. All those are my favorite molecules. So.
Great. That's definitely a really, really great selection of molecules right there. What are you most excited about in drug discovery today, Jagat?
Again, when I think about look back for 25 years, I think we learned a lot, and we only hit the tip of iceberg. And again, we have definitely brought some of the greatest drugs to help the cancer patients. But if you ask me, did we cure the cancer? Probably not yet. So yes, certain population are getting benefited, but still majority, I think, especially solid tumors, still untouched. And there's a lot to do for all of us. And every day I'm excited about it. How can we build next generation therapeutics? And that's what motivates me. So.
Amazing. And we're really looking forward to hear more about some of your ideas with ADCs to tackle those exact questions today. If you could go back in time to when you started your career and give yourself some career advice, what would you say to yourself or to the younger folks here in the audience today?
I think, again, this is something I always do this, mentor the younger folks. In fact, for that reason, I started an organization called Science Gurus, which is career advice for high schoolers, basically. As we said, we just completed our drug discovery workshop last week. And we have done about 400 people so far, past 13 years. So, and what I do, introduce them translational science very early on. So again, we need smart people in biology. And of course, IT industry and all other people attract automatically, but biology needs smartest people. And I think introducing translational discovery, translational science very early on to these people probably make their choices, and hopefully we can attract some of these young scientists to the industry as well, take on their career. I think that's my advice. And if anyone wants to join the translation science, I think PhD and going to postdoc in the industry, that's what I recommend. That's what I've done it, even though my postdoc in the Genentech didn't happen with planned, but happened accidentally. But there are a lot of companies offering postdoc opportunities in the industry. I think joining industry as a postdoc, if they want to continue a career in industry, is the best way to go, so that you can sort of view whether this is what you want to do or not. I think that's the advice I highly encourage for people to consider. Industry postdoc is not popular, but it is getting popular. That's what I would say.
Awesome. Yeah, thank you, Jagat. And yeah, last but not least, what do you like most about Drug Hunter?
Webinar. That's why we are here. And I think it's a great way to exchange the ideas and thoughts to the rest of the world. And I think these virtual webinars are becoming more popular ever since we hit the COVID. I think this COVID brought something is close to the world is through the webinars and virtual meetings. And it is definitely, I thank you for doing this. Actually, it's really, really good. So I definitely like the webinar part, most excited about it. Thank you.
Awesome. Yeah, thank you. Greatly appreciated. Yeah, before we get started, I want to show really brief one new feature that we recently introduced at Drug Hunter, which is here. And this is our new estimated human dose calculator. So the way you get there is you go to resources, you go to calculators, and then you see a page of all the calculators that we have here. And the latest one that we have is the estimated human dose calculator. So you can click that right here. And then you can basically use that to estimate your human dose. So you can, for example, put in your IC50 here. You can put in a molecular weight. Let's just put in a number. We can put in plasma protein binding in an animal species, plasma protein binding that we measured in human. So let's just put in a few hypothetical numbers. We want to put in a volume of distribution, and we want to put in an oral bioavailability here. So let's put in 74%, and then a predicted clearance. Put in something like this, and you will get an estimated human dose here. So if you have a compound with those parameters, your estimated human dose for 70 kilogram human would be 65 milligrams dosed once a day. You can also toggle here between BID dosing, for example. Then you have a bit of a lower BID dose. You see the overall dose becomes lower in 44 mg, and the individual dose would be 22 mg. You also get the mg per kg in case your human is not weighing 70 kilograms. So all of this information is available here for you, and we'd encourage you to try and use that. And now, without further ado, I'll hand it over to Jagat for his presentation on multifunctional ADCs in oncology. Is it a fact or is it fiction? We'll learn the answer today. Jagat, the floor is yours.
Again, thank you for Drug Hunter for giving this opportunity to present some of the thoughts, but I just wanted to put one disclaimer because the talk that I will be giving pretty much my 25-year journey condensed into 30 minutes talk. And the views that I'm presenting, my own personal views and opinions, not necessarily views from Arvind. I just want to make the disclaimer. So. And with that, what I did, I sort of divided my talk into 3 buckets. And first portion about more like a general introduction to ADCs, but we talk about what we learned the first generation ADCs, and then how we took those lessons and moved on to develop second generation ADCs. And some of those second generation ADC, I'm going to give important highlights. I'm going to put it. Then last but not least, we talk about all these lessons from first generation, second generation, how we implementing and developing next generation ADCs. That's Arvind as an example, and I'm sure other people are doing as well. And but today focus will be Arvind's point of view at the last section. With that, and again, I'm sure all the people joined the meeting and aware what is an ADC. So again, in principle, ADC is a 3-component molecule. We have an antibody, which a payload is attached to antibody by a linker. So if someone asks which is important, I would say all 3 are important equally. So antibody, linker, and payload, they all play a very important role because, again, as an antibody coming from large molecule world, payload is coming from small molecule world, you are marrying the benefits of both. You're also marrying the problems of both too. So you have to think through, when you're developing an ADC, you have to think through, you have to think as a small molecule drug discovery person and also large molecule drug discovery person. So you have to bring in both the challenges as well as advantages. So again, why ADCs are exciting? The concept is not new. It's been 100-year-old concept. I'll tell about the history in a minute. But why ADC is exciting is, if you think about chemo, again, everyone, pretty much every cancer patient receives the chemo, and they go through this toxicity effect because chemo doesn't differentiate between normal cell versus cancer cells, so therefore it kills all cells, so therefore you don't have any window. But when you attach a chemo to an antibody is more like a guided, this ADC is more like a guided missile where antibody guides and missile is the chemo. So because of that, ADC binds the tumor cell surface with the tumor antigen present on the surface and gets through internalization, and then in the lysosome, the antibody gets degraded and linker cleaved and payload release, and release payloads can act on its mechanism of action. So that's for all this concept, how it binds, how it gets inside the cell, how it gets cleaved, and release the metabolite or payload, how does it act? All these are a very important part of the ADC drug development. So again, as I said, it's a 100-year-old concept. Paul Ehrlich is the one first coined this term called magic bullet for ADCs. But as field moved on, there are many important discoveries happened, both antibody side on the linker payload side. And I think some of these most important inventions, so, how people use methotrexate first to linking antibody, to how people develop an antibody through hybridoma technology, then process called humanization. All these are very important. I think the most important discoveries in ADCs are start with the first FDA approved from Mylotarg to Adcetris approval in 2011 and Ketsera from 2013. These are the one actually ignited the field again for a very long time and kept scientists like me enthused about this field even today because of these early discoveries, what excited all of us basically. So when you think about ADCs, you think about not just three things. When you think about, you think at least 6 different factors and antigen. Why given antigen actually overexpressed in tumors or minimally expressed in normal, whether antigen internalizes, whether antigen sheds. These are all important factors to think about it. Then you think about antibody, epitope, affinity, specificity, internalization, functionality, and many other things. I will keep coming to some of these things throughout my presentation. Then think about conjugation technologies, whether you're making specific or non-specific conjugation technology. The linkers, cleavable, non-cleavable, hydrophilicity, stability, plasma, bystander effect. The payload, again, multiple attributes for the payload, and the indication itself, and given indication, not single payload will work. And you have to diversify the payload for all the different indications that you want to work on. So these are all important features. When you think about ADC drug development, you have to think through all these things. And just wanted to have a quick summary of clinical landscape, what we learned today. And again, as I said, what we thought window of ADC therapeutic window is very broad, but happened to be a little bit narrower than what we expected, partly because, this is as of March, probably the number changed as of today, 466 ADCs that progressed into clinic, and of which 14 approved, 315 active, 137 discontinued and inactive. So if you look at the approval rate versus discontinued, about 90% failures we are seeing it. And why can we make it better, that number? And how can we make that better? I think this is what all the ADC field, the scientists are continue to think about it. But the challenge has been, we thought we spend significant amount of time trying to improve the safety, that is maximum tolerated dose. In somewhat, I think I must say we failed, partly because this off-target toxicity is the one limiting, which is happening through macropinocytosis. I'll be talking more about it. So, and again, if you combine this 466 ADC, how are they distributed among the tumor antigens that we selected, thanks to Onsen Wade Beacon ADC database that we have access to. And if you can see, there is a handful of targets, about 10 of them pretty much cover the majority of these targets. And again, probably too many ADCs that we have been transitioning for a given target. So something to think about as an ADC scientist. We all, we need to be probably cognizant of number of molecules for a given target that we are transitioning, something to think about it. And likewise, linker payload, predominantly first 20 years, we focused on 2 different kind of linker payload. One is auristatin family, other one is metanatin family. Then PBDs came, but Topo one pretty much after Enhertu success, Topo one pretty much took over entire ADC field. In fact, last 18 months, we have almost 80 Topo one payload ADCs went into the clinic. And what are the targets being used? Same targets. Whatever failed targets, we are repurposing and using Topo. Would that work? So time will tell. But again, I think, we are focusing too much on Topo. And I think other payload need equal attention and something to think about it as well. So I think, again, I'm going to focus more on MMA ADCs, partly because we learned a lot, and they are almost 40% of the approved ADCs belong to MMA ADCs. And if you look at the data on MTD versus MED on that, and what is the striking or important feature that we notice, and if you look at all the approved ADCs such as Polivy, etc., Tivdak, and Padcev, and among all these listed here, only Padcev use a different dosing regimen and dosing schedule. So remaining all Q3-week dosing. And if you pay attention to that, MTD is pretty much standard, 2 to 2.4, all the ADCs. But what is interesting is the MED. If you look at the approved drug, MED, they are close to 1 mg per kg. The one discontinued MED, they're close to MTD, or they don't have any efficacy, noticeable efficacy, much below the MTD. So in other words, if you can somehow figure out engineer ADCs to improve MED by 2-fold, pretty much you can see the failed drug to the success drugs, basically. So in bottom line is, we are stuck with MED. How can we lower the MED is the question that I think we all should think about and ask about it. So it is MTD is MTD. You can't, because of penocytosis, you can't really improve a lot on MTD, but can we improve MED? Can we improve the efficacy? How can we do that? I think that's the question we all should ask. So I think some of these lessons we learned in the first generation ADCs are being implemented in the second generation ADCs. One is payload diversification happened a lot. So again, as I said in the beginning, orostatins, methansins are the one got major attention in the first generation, but second generation, obviously, we went either extreme left side using more potent payloads, or extreme right, the less potent payloads such as topo. So we learned a lot through that. And of course, because of that opportunity, we are able to either lower the dose or increase the dose. So we'll learn some of those examples. And some of the choices is that efficacy driven. And we also learned about engineering antibodies, engineering linkers, and decreasing hydrophobicity of the linker payload. So multiple things were implemented during second generation. Quickly summarize some of those, and what are the lessons that we learned is that I would like to focus in the next few minutes. So again, the most exciting example of all this, Kadcyla versus Enhertu. And if you remember those two molecules, what is really different? So target is same, HER2, and antibody use is same, trastuzumab or Herceptin. And only thing differs between both is the linker payload. And Kadcyla uses non-cleavable linker, and Enhertu uses a cleavable linker. And Kadcyla uses a tubulin inhibitor, methansine, and Enhertu uses a topoisomerase 1 inhibitor, topo 1 inhibitor. So, but Kadcyla, what surprised us, Kadcyla, is the preclinical data to the clinical data. Kadcyla, if those who remember Kadcyla preclinical data, it's a 30 mg per kg in the monkeys in the Kadcyla. And we were expecting about 10 mg in the human MTD, but we landed with 3.2, 3.4, actually 3.6 mg per kg in the Kadcyla clinical dose. So what changed us is the surprising thrombocytopenia, which actually gave a DLT in the clinic, and which didn't let us dose beyond that 3.6 mg per kg. And whereas when we changed, when the Daiichi changed the linker payload to methansine to topoisomerase inhibitor class, so they didn't see thrombocytopenia, and they saw instead a lung tox, which is about ILD, interstitial lung disease, about 10 mg per kg. So the MTD for Enhertu is about 8.4 mg per kg, and MED is 3.2 mg per kg. So what does it really bring it? So it completely brings a different dose range, which is in the range of 6 to 8 mg per kg, and interestingly, that's where you see Herceptin function. So in other words, Enhertu uses Herceptin function blocking activity as well as ADC activity. Kadcyla cannot use Herceptin function blocking activity because the 3.6 mg per kg Herceptin function is pretty much negligible. So I think that's an advantage by using a low potency payload class. You are moving your dosing from lower to higher, and which captures the antibody function. So it's, in other words, Enhertu is a dual acting ADC, I would call perception function blocking as well as ADC activity. That's something to note. And when you're thinking about ADC design, how can we maximize antibody role here? Okay, not just guided delivery purpose, but also can we utilize function blocking? And then obviously at higher doses, you have better exposure. So multiple things went into right direction for Enhertu where it didn't it didn't happen. So then think about PBDs, and this is not the only example I would like to highlight. And PBD, there was the era of 2010 to 2020, there was a PBD era, and it went up and went down, multiple different reasons, but still it's a payload that I consider is very important contribution it made in the ADC field. And for example, something like AML, you can't use maytansines, and in other words, antitubulin inhibitors. And why? Because AML has two kinds of cell population: leukemic stem cells and bulk tumor, and blast cells, leukemic blast and leukemic stem cells. Leukemic stem cells, they are not proliferative type, but leukemic blasts are highly proliferative. So you need to have a payload that actually kills the quiescent cells and highly proliferative cell population. That's where PBD or calciamycin and any of the DNA-damaging payload really work very well, because they can kill LSC and as well as blast cells. And again, the choice of the target is also very important for AML, whether you use something like CD33 or not. And CD33 expressed in HSC, hematopoietic stem cell, but also expressed in LSC, but expressed in blast. Whereas other markers such as CLL1 and a few others, and do not express in HSC, but express in LSC and blast. This is an example that we published several years ago, 2018, and sort of talks about all these things. So target expression and payload selection is very, very important when you're thinking about ADC. Then I think, again, this is my favorite part of the story, is engineering antibody and making site-specific ADCs. How did that change the industry a completely different direction? Before we worked at Genentech and site-specific ADC, pretty much everyone was using in Kadcyla type lysine-based conjugation coined by Immunogen, and then cysteine-based conjugation coined by CR Genetics, etc. So both have, again, conjugate to antibody specifically, but they conjugate different sites. And Kadcyla used almost up to anywhere from 0 to 20 lysine, and etc. is used anywhere from 0 to 8 cysteines, and that gives heterogeneous mixture of molecule and all that. But at Genentech, when I was Genentech, we invented a technology called Thiamab, and after that paper, famous 2008 Nature Biotech paper, the many people came up with so many different versions of site-specific ADCs, and this is in fact, a short list. The list goes on now, 30 to 40 different flavors that one can have it, basically. But again, the way we think about site-specific ADCs is changed ever since this 2008 Nature Biotech paper came. Why? Because even we are able to dissect where to put this cysteine is also makes a big deal. And we, again, another 2012 Nature Biotech paper, we published this and said that the location does matter. And because how when you attach the maleimide to the cysteine, the succinimide ring opening versus exchanging to cysteine glutathione albumin present in the plasma can contribute to the efficacy, stability of the ADC, and sequentially to the safety of the ADC. So this is very important, and where you put it. And after this discovery, the ring opening, people went on to synthesize linkers, engineer linkers to trigger the link opening, and many other things happened. I think, again, it's made an important contribution to the field of ADCs, how we design. Now pretty much every company is working on site-specific ADCs. And I think next important thing we learned a lot about how can we decrease the hydrophobicity of the ADC. Again, this came from CR Genetics, and they designed this branch linker, linear linker, adding PEG in the linear or adding PEG in the branch. And I think opened completely new way of thinking. How can we make hydrophilic ADCs? And again, here you can see this is again Nature Biotech paper published in 2015, Peter Sender and colleagues, and very, very elegant part of the data, and how you put the peg position spacer in the branch versus linear. Linear remains hydrophobic, but branch gives hydrophilic because that peg is probably wrapping around the payload and shielding the hydrophobicity and making it hydrophilic. And that contributes to improving PK and efficacy, and obviously limitation of liver absorption and liver clearance and all that is limited when you make it hydrophilic. And this sort of triggered many different people coming with many different hydrophilic versions of linker payload that we are seeing today. So again, this very, very important lesson that we learned throughout. And if I can summarize some of these in the form of safety that we learned, I always like this analogy of ADC safety with an onion bulb. Why? Because we all see onion. Everyone, all of us probably seen onion, and you cut open the onion, and you see many different layers you see. So I see ADC safety is more like onion bulb analogy. I think if I take MMA an example, the top layer of ADC safety is called neutropenia, which we saw in the clinic 2.4 mg/kg, and that's what MTD, pretty much all MMA ADCs that we've seen went into clinic with noticed neutropenia is the DLT, and that's typically 2.4 mg/kg if you use the random cysteine conjugation method. If you use site-specific, this pretty much disappears, and neutropenia, basically site-specific ADCs eliminate the neutropenia layer. Then you hit ocular tox. That is 4 to 5 mg/kg range. And that's what first generation versus second generation MMAE. First generation didn't use site-specific ADC. Second generation using site-specific ADC, we are seeing ocular tox. But what we interestingly we found is a term called lung tox. The first time we noticed when we were working with site-specific ADCs and in the monkeys, and that we have seen anywhere from 10 and above, and 15 is more prominent, 10 is small lesions. So, and we have seen that. So at that time, we didn't know, this is the first time we heard about lung tox in ADC. Everyone was pretty much nervous about it. But ever since topo ADCs came into the picture, every topo ADC, that especially the one which can be dosed higher, above 6 mg/kg, started seeing ILDs. So lung tox is always there, but it's hiding behind ocular tox, basically. So in MMA ADC, or metansins, or auristatin, MMAF, you see ocular tox around 4 to 5 mg/kg, or 6, more prominent. But lung tox was hiding behind that layer. Once you remove that layer, you hit the lung tox, whether it doesn't matter MMA or topo, basically. So these are the toxicity layers. They're always there. You try to eliminate one, you are going to hit with another one. So these are pretty much driven through, except neutropenia, everything else driven through macropinocytosis and large molecule uptake into the tissues and cells probably. But neutropenia is driven directly released payload attacking the white blood cells population. So again, these are the toxicity that we have been encountering, and it's what it is. And how can we get away, how can we stay away from these toxicity layers? What can we do is the question that we all need to look for. So again, another quick summary on this MTD doses. If you put MTD doses on the payload, they're pretty much linear. If you go with high potency payloads such as PBD, the MTD range was 0.1 to 0.5, depending on the attribute that you look at it. If you look at metansins, auristatins, and the MTD dose is the potency is sub-nanomolar, but MTD is 2 to 6 mg/kg, depending on the target and many other things. But if you go to low potency payloads such as topo, and it ranged anywhere from actually 3. I should correct it. Some of the exoticam payload now they're seeing around 2.5 to 3. So anywhere from 3 to 15 mg/kg, depending on the payload potency that you choose. So it's pretty linear. If you look at payload potency and MTD in the clinic, the pretty much a linear relationship. So you decide which payload you want to pick. What is the dose range you want? Pretty much it's pretty linear. If you look at all hundreds of studies that we have done, they're pretty much linear. So something to think about what dose range one would want to choose it. So I think for this part of the story, I would like to quickly summarize, thinking about, again, all my lessons learned through pretty much an MMA ADCs. If you think about MMA ADCs that we worked on so far, there is a delicate balance between ADC efficacy and safety. And I would, and a very good analogy here, if you think about this cartoon, the balls, an elephant sitting on a ball, I think elephant is thinking that it's controlling the ball, but ball is thinking it's controlling the elephant, actually. So if you think about antibody as an elephant, linker payload is the ball, now you can see who is controlling whom. Ideally, you want to see antibody controlling the linker payload, because antibody role is not been exploited a lot. Antibody should do a lot more than just delivery. So how can we exploit antibody engineering capability to broaden the therapeutic window is the question that we all should think about it. And that's what Arvik is actually founded in that basis, to see, can we engineer antibodies to make it broaden the therapeutic window? How can we do that? So with that, the problem statement is very simple. And if you think about approved ADC, the target copy number is pretty much 100,000 to 1 million. And if you think about HER2, TROP2, Nectin-4, FR alpha tissue factor, I think the DLL3 is the only one exception here, but pretty much most of the ADCs fall in this high copy number target. And most of the failed ADCs, if you look at it, they fall in this low copy number target, 50,000 or below. So how can we make this failed ADC fall in the low copy number target, make it successful? What kind of technology one would need to use it? So again, I've been telling about is it's not about safety. Safety is great if you can improve it, but I think huge opportunity for us is improving efficacy. How can you improve efficacy? How can you improve your MBD dose? That's what Arvik is actually positioning itself. Is we are mostly spending our energy to improve the MBD dose. And of course, in that process, if we can improve MTD, great, but that's what is our Arvik focus is the thing. So with that, and myself and Dr. Dev Sidhu were the co-founders of the company. We started this 3.5 years ago, and we have a small team, but very effective team with a lot of industry experience, and with we can take idea from IND level, and we are super excited to have this team. But most importantly, we have a very knowledgeable advisory board who are veterans in the field of drug discovery, and also the board who was guiding us all along. We're super excited to have this team, and all these profiles on the website, you can take a look at it. But this is what I think about when I think about as a human being. We have two hands, right? And ADC has two hands too, just like us. But when you want to do multitasking, you must have thought, I wish I had two more hands. I'm sure the thought would have gone to your head. Like, Swiss Army knife or 4-in-1 ballpoint pen, multi-arms, multi-skills, basically. I think that's what we are thinking about. A cartoon that I'm depicting here is a depiction purpose only, not exactly the cartoon that we use. But the idea is, can antibody have 4 arms? And 4 arms with 2 or 3 or 4 different functions. Then 5th function is coming from linker payload, or 6th function coming from linker payload. So you can have dual payload too, or whatever, right? The idea is, how can antibody have more arms, and how can we utilize it? I think this is the question we're trying to address ever since we started Arvik. And for that, we use first example is, can we engineer the approved drugs into single biologic? The answer is yes. We engineered Herceptin, cetuximab, Prozeta, and another EGFR binder, 4-in-1, basically. And of course, a lot of work went into it to ask this question, but elegantly this was engineered in a co-founder's lab, the Sidhu's lab. And the question about, when we engineer such a complex molecule, can you manufacture it? I think that's the other important thing is, yes, the answer is yes. Single-step purification, basically, simply take these 3 constructs, 1 light chain to heavy chain, put in the test tube and transfer, throw it on proteinuria, you will get pure 95% yield. We have done several thousand times this experiment, and it works beautifully. But so what, right? You engineered multifunctional antibody, you produced it, manufacturing is not an issue, but the question is, can you test functionality? The answer is yes. So when we have a MUDA, actually what we are looking at is our phosphorylation. Monospecific antibodies don't block the phosphorylation in this trastuzumab-resistant cancer cell line, whereas a typical bispecific partially block, but multi-specific MUDA actually eliminates that phosphorylation completely. So you do see an advantage when you bring different functionality to the same biologic. Then we ask the question, can we convert this as an ADC? And when you bring a multifunctional antibody, does it trigger internalization? This is antibody-mediated internalization. So we're talking about antigen internalization. Typically, antigen drives the internalization, antibodies don't. But in this case, antibody drives the internalization. So you have three molecules here. A control MUDA, which doesn't bind cells. Then you have trastuzumab, and then you have a MUDA which binds cells in multiple different sites. You can see MUDA triggers internalization robustly, and control doesn't do, and whereas Herceptin does normally what it does. So what you're seeing between cyan versus green, antibody-driven internalization. This is missing in the ADC field. We have not really cashed on this antibody-driven internalization significantly, and this is what multifunctionality can bring it. Then we asked a question, how can we build ADCs through multifunctionality? So here you're looking at is two monospecific ADCs in the green and the purple. Then we combine these into a 4-arm multifunctional ADC, and then tested in these are three independent projects, and tested the activity of the ADC potency. And you can see in the breast example, we have a MUDA has activity over 23x on one ADC, 40x is the second ADC. These are both monospecific. And lung, we have 51-fold and 2-fold in improvement over monospecific. And in prostate, we have 14-fold, 11-fold improvement over. So we have tested this multiple times, and we are excited about this, combining the epitopes, combining the paratopes, and combining the different antibody functionality and they improve the activity. One question comes when you make novel antibodies with different shapes and all that, how does the PK look like? The PK of our MUDA molecule is similar to the Herceptin or pertuzumab, or trastuzumab pertuzumab. In fact, in mice as well as in monkeys, we tested and similar PK. So at least our novel engineered antibody PK is not compromised with similar PK, which is we are super excited. We also, again, interest of time, not going to go into the linker payload side. We do have proprietary linker payload with the different mechanism of action we engineered with the hydrophilic functionality and all that. Hopefully some other day, some other time, we can talk about that. And I think when we think about ADCs, we don't think about one attribute, we think about 6 of them. How can you increase the copy number? And you can't increase literally a tumor antigen copy number, but you can increase the copy number by combining the functionality. You can improve the internalization. I showed an example, increase the valency, increase the binding architecture and hydrophilic linkers, diversifying payload. When we design ADC, we think all these 6 different angles to truly develop a next generation ADC. I think we went this idea from all the way from a concept to all the way to PK and safety in monkeys, and we have confirmed that we improved the therapeutic index at least by 10-fold. Super excited about it, this platform, and we have thought about it, and how does the MUDAs work actually? So typical monospecific ADCs bind one tumor antigen and kill it, but in the case of MUDA, actually we have incorporating multiple binding sites in the molecule. It kills basically target A, target B, target AB. So basically it's OR-gate. Basically, in all 3 scenarios, it works beautifully. So again, we as a company were super excited about this platform, and we have 20+ drugs that in the pipeline that we open to collaboration. Anyone is interested, and you're most welcome to come and talk to us. And with that, thank you, and look forward to the Q&A session.
Thank you so much, Jagat. That was very, very impressive data. The audience also mentioned beautiful data, and thanks for sharing this with us today. Yeah, really exciting to see how they can be such a big shift with the MUTA antibodies. And I found the layers of the onion also really, really compelling. How you first have neutropenia, then ocular tox, and then this lung toxicity. So yeah, really educating for me as well as somebody who's not super familiar with ADCs. But yeah, let's go to the audience questions here. So the first question comes from Bill Mallet, and he's saying, hypothetically, oh, you know him, Bill, I'm assuming. Bill also is saying that, he's thanking you for sharing the beautiful data.
Bill and I worked together. That famous 2008 Nature Biotech paper, we co-corresponding author, and yes, we worked together a lot. All
Awesome,
right.
awesome. So yeah, his question is hypothetically, antigen independent off targets of ADC toxicity can be modulated by various antibody optimization strategies. For example, FC silencing, engineering of site selective conjugation, tuning of biophysical properties, for example, thermal stability and aggregation. But do you think that FC engineering to prevent FCGR binding is a promising strategy to improve systemic safety for ADCs?
The bill, I think you know this as much as I do, and I think that's a debatable question. And I've seen examples. Again, we have not seen direct clinical evidence anyone doing FC engineered FC gamma receptor deleted function ADC versus normal head to head. Unfortunately, you can't do that kind of study in the clinically, but you can do preclinically. But preclinical models are a little bit challenging. I don't. First of all, FC gamma having FC gamma receptor function doesn't contribute to ADC at all. So ADC activity. So getting rid of it probably is the right way to do it. But do I have the answer? I don't have the answer. There is no experimental evidence to confirm this versus that. It's anyone's toss. If you ask me, I would get rid of it because it doesn't contribute to ADC activity at all. At least if it improves great. But we have unfortunately don't have a direct evidence to confirm this way or that way, basically.
Yeah, thank you. And you mentioned about the toxicity. Huang is wondering, you mentioned there's lung toxicity in non-human primates. Does that also correlate with the lung toxicity in the clinic, or are there any other approaches to de-risk the lung toxicity preclinically?
Unfortunately, there is a black box to many people. I don't think preclinical lung tox and monkey tox was directly looked at the lung tox that's seen in the patients. Anyone looked at carefully. So again, I'm not seeing that kind of study directly, and I can't really answer that whether they're same or different. But these are what we have seen is the microscopic lung lesions. So. And maybe a pathologist can answer. I'm not an expert to answer whether they're same lesions or not. But what we have noticed, we have tried all different ways to eliminate it. We couldn't do it. So we at the end, we concluded it's a macropinocytosis-driven mechanisms and off-target toxicity. We can't really eliminate it by any means. So you just don't go into that dose range, basically. For example, we have seen lung lesions with MMA ADCs about 10 mg per kg. In monkeys, but in theory, we can never go that kind of dose in the people. Only I think topo payload class enabled that. But MMA or starting, they hit ocular tox. They never got to see lung tox in the clinic because ocular tox comes before lung tox. Therefore, it's hard to compare MMA-driven lung tox with topo-driven lung tox lesions or same or not. I'm not a pathologist, so therefore I don't know anyone looked at carefully.
Yeah, that's a great answer, I think. Amol Iwale is wondering about a specific technology for cold tumors. So he says they are binding linkers with specific cytokines that turn cold tumors into hot tumors. How would you think about this type of strategy for the next generation of protein-protein conjugation? And are there any thoughts that you have about this particular technology?
Oh, absolutely. I love it. Any way you can arm the antibody-drug conjugate to a different functional modality, it's fantastic idea. Go for it. If you have access to that kind of technology, yes, we would love to explore that. Absolutely. I like the idea.
Great. And how about there's a question from an anonymous attendee about antibody degrader conjugates. What's your opinion on antibody degrader conjugates?
In the ADC field itself, there's so many things to do it. And people are already working on antibody ADC, degrader linker payload options. Again, it's a novel idea. You're using different mechanism of action and all that. But think about it. Many other things to worry about it. That's not the only thing you need to worry about, the degrader function. So if we can handle all those attributes and add other additional layer of complexity, yeah, go for it. Absolutely. And I'm always have a positive views on diversifying the payload mechanism of action, and degrader can certainly bring a different payload mechanism of action. And again, that's something we're not focusing at Arvic, but I'm sure the other people are already embarking on the idea. We will have, probably time will tell whether that was a great addition or not. So.
Cool. And then how do you think about, Sean Gao was asking that, how do you think about multi-specificity leading to increased internalization versus increased on-target off-tumor tox in healthy tissues?
That's all about target selection. If you have a dirty target expressing a normal cell, it is going to kill it. So look for target. They are minimally expressed in normal tissues, maximally expressed in the cancer tissues. Otherwise, you will increase on-target toxicity too. Obviously, there is no brainer on that. So it's all about target selection criteria, how you will apply, how you select how you pair it. It's all about that. And that's proprietary for every company, and no one is going to tell you their magic. So yes, if you increase the multi-specific, but that liability you will have in the monospecific ADCs too. For example, TROP2, right? And TROP2 you put on MMAE, patients died. But same TROP2 you put in topo, patients did not die. So if that you combine with the target payload, linker payload selection, and that's what it makes sense. So it's about target selection, linker payload selection, how you combine this is combination of all this complexity. So there is no straightforward answer to it. It's about you have to think about all this, how you want to combine it, essentially.
Makes sense. And then Sampath is wondering about your thoughts on multiple payloads for an ADC.
Yeah, I know. Again, I don't know about multiple payload, but dual payload is getting a lot of attention. This is the only thing I can comment on it. I don't know whether we have successfully handled the single payload yet. Carefully, okay? If you're thinking of dual payload, I would say great. You know, if you can combine, synergize, manage the toxicity, go for it, okay? But you add another payload, you bring a different toxicity parameter into it. If you can carefully synergize that, I think good luck, absolutely. But again, time will tell. I think just dual payload started getting a lot of attention, and I think in our view, I think combining the target is probably a lot more exciting. Combining the payload, it requires a lot more thinking process. If you can successfully do it, great.
Yeah, makes sense. And then another question from Bill. How can the MUTA architecture enhance ADC delivery into fibrotic, poorly vascularized solid tumors?
Bill, I wish I have an answer. I will, when I know the answer, I'll call you and tell you. But I don't, we have not far yet to address all those. We have so many things on our hand. We can only do so much. So, but definitely the day I have an answer, I'll call you and tell.
Awesome. Great. Another question about the toxicity. So the ocular tox is usually attributed to the MMAE, while the ILD, so I guess the lung toxicity is attributed to topo. Are there actually layered or different types of payload risks?
No. MMA, ocular tox is not just MMA. Ocular tox
Okay.
in MMA, ocular in MMAF, ocular tox in metalsins, all tubulin inhibitors actually have ocular tox. Okay. So coming back, I just told you, we have seen lung tox also in MMA ADCs in the preclinical models because we don't see ocular tox in the preclinical model. We bypass the layer and fit the lung tox. So I think these are always hiding behind the layer. They're always there. It just topo doesn't have ocular tox, so it straight went into lung tox probably. Whether they have similar lung lesions or not, I don't know. I can't comment on it. But in my view, these layers always there. So.
Got it. And then Harch OE is wondering, for the MUTA ADCs, are you using site-specific conjugation? And can you comment on the DARs that you found most effective?
That is proprietary. I cannot comment. What I can say, you can use any DAR of your choice. You can use DAR2, DAR4, DAR8, or whatever you want to conjugate. This is like just like any other antibody, right? So Muda platform, I have an opportunity to conjugate how many DAR you want. So DAR is not again, DAR is a personal choice. Everyone has their own reasons to use particular DAR. If you use a high potency payload, they want to use DAR1, DAR2. If you want to use low potency payload, they want to use DAR8. So it's a personal, individual company choice based on the linker payload they have. But in my view, DAR is not a big deal. DAR is a number, and there is nothing like a favorite DAR here. It's all about which linker payload you are using, what DAR you want to pick it. So it's linear. If you use DAR2 or DAR4, you tested DAR4 as twofold more toxicity, twofold better efficacy versus DAR2, but it's pretty much linear relationship between DAR2 versus DAR4 versus DAR8. So what you want to pick is up to individual choice and individual drug and how you pick the linker payload, what kind of dose range you want in the clinic. There are multiple parameters to pick the right number of DAR, but doesn't really improve the therapeutic window. Therapeutic window is static between DAR2, DAR4, DAR8. Yes.
Got it. Yeah. And then Fernando Padilla is wondering, the multiple targeting approach with, I guess, Muta might increase the risk of targeting healthy cells. How are you addressing that issue?
I answered that question already. Basically, I said it's all about target pair selection. How you do it, it is as simple as that. And if you pick a target that expresses abundantly in the normal tissues, obviously you're going to. But that is the case with the Yamuta, where there are monospecific ADC too. You have to wonder about that. So yes, it's all about how you align your target pairs. It's very, very important.
Got it. And then I guess we have time for one more last question, although there are a lot of unanswered questions. So I guess it's been a very great talk, and a lot of people are very excited about the data. Even questions about the phase 3 data that I'm sure you probably cannot talk about. So last question is in the ADCs field, is this still driven by empirical designs, or is there enough knowledge and data to drive rational ADC designs?
It is rational ADC design. The era of empirical, no. Actually, ADC has to be rational design. So unfortunately, that's what I worry about it when I see 80 topo ADCs went into the clinic. That is not a rational design, unfortunately. So everyone has their own reasons why they went into clinic, but just because they failed with MMA ADCs, they switched to topo because they know how to work, they're going to work, probably not. So we got to spend more time and energy in rational design and thinking. I think we can improve the success rates from 10% to probably 50% if we can do rational design, apply more carefully and knowledge, and use the preclinical data to make a judgment whether this molecule deserves to go to clinic or not. I think that's what we are not paying attention to. Preclinical data, there's a lot of knowledge. There are 1,000 ADC papers actually published every year. Past 10 years I've been watching. Basically, I pretty much look at all of them. And there's a lot of knowledge out there. I highly encourage everyone to read it, please, because there's so much information already there. If you apply all that knowledge and scientific basis and design rationally, I think we can do a lot better job as ADC community. So yes, my personal preference, rational. That's what we apply at Arvic, rational design. I don't know how others do it, but rational design is very, very important for ADC success.
Awesome. Thank you so much, Daggers. That was an amazing presentation, and thank you also for sticking around to answer so many audience questions. So yeah, round of applause for Jagat again, virtual.
Thank you, Dennis. Thank you, Drug Hunter. I'm super excited to share some of the thoughts, my personal experience, and also a little bit of Arvic story. And hopefully we get to hear more and more about Arvic stories in coming months and years. And we are super excited to be part of this ADC community. Thank you.
Awesome. Thank you so much, Daggett. And yeah, we will be back here on August 28th, so in 2 weeks, with Mark Murko. And Mark will talk about understanding how drugs interact with anti-targets, the avoidome. So that is an interesting topic as well. And with that, we wish everybody a wonderful rest of your day and wonderful rest of your week. Thank you again, everybody, and thank you, Jagath.